and high basal activity in the regions that did not give associated response. Thus, using AMPK activity reporter, we are seeing spatial differences in the stimulated responses of AMPK. So it's evident that key information is present in these spatial compartments of cells, and it's just as important as it is to monitor these signaling events. It's just as important to perturb these signaling events, and site-specifically perturb these signaling events. Their small molecules can be specific, and they have good temporal control, but spatial control is hard to achieve. And current tools for targeted perturbation include chemically as well as optically inducible switches. Over here, I have an example of a rapamycin-induced heterodimerization strategy where the two proteins do not interact with each other, do not undergo heterodimerization until you introduce a small chemical called rapamycin, and this causes the heterodimerization of the two protein fragments. So just as we are, there's a plethora of tools to monitor signaling dynamics in cells, but we're kind of lacking a little bit in the perturbation department. So so we wanted to develop a tool for targeting, so we wanted to develop a tool that could kind of facilitate targeted perturbation and thus expand the toolkit to complement these existing methodologies. So I've so far shown you an example of how a unimolecular kinase dependent switch was employed to monitor signaling dynamics. And a graduate student in our lab, very talented graduate student in our lab, Katie Herbst, Dr. Katie Herbst now, she showed that when this molecular switch is broken into two different independent fragments, a kinase dependent phosphorylation event can induce the heterodimerization of these two components of the switch. So this now becomes analogous to the rapamycin system I introduced in the previous slide. And then we wanted to use this switch to kind of regulate cellular events as a functional output. To do so, we targeted the substrate part of the switch to the plasma membrane, and then to the phosphoamino binding domain, we coupled an effector, a signaling unit, some signaling molecule that served as the effector unit. And upon kinase activation, this the phosphorylation will lead to the binding of the phosphoamino binding to the substrate, binding domain to the substrate, resulting in the localization of the signaling unit close to its substrates of the plasma membrane. And furthermore, because these phosphorylation events are susceptible to antagonistic activity by phosphatases, this gives us a handle on reversibility. And if I may, like I think of it as a toggle. So basically, you can switch on and off the phosphorylation event, and that can help you relocalize signaling molecules. So as a proof of principle, we used a switch that was sensitive to protein kinase A, and the phosphoamino binding domain used in this switch is a fork head associated domain 1, and coupled to that domain is a yeast inositol phosphatase, which catalyzes the hydrolysis of PI4P to PI4-5P2 to PI4P. And the PKA specific substrate was targeted to the plasma membrane using a plasma membrane targeting motif. The hydrolysis, the PIP2 levels were monitored using the pH domain of phospholipase C. And all these three constructs were tagged with fluorescent proteins so we could monitor the expression levels as well as the localization of these different chimeric proteins. And in an unstimulated cell, when all these three proteins are expressed in it, you can see the localization of the FHA1 domain is it's just cytosolic, while the pH domain here is mostly associated with the plasma membrane. This is where most PIP2 levels are. So upon maximal activation of PKA using using forskolin plus IBMX, we see a nice decrease in the cellular cytoplasmic intensity of cerulean, and because this results in the translocation of M54 to the plasma membrane, you see the pH, and it results in HIP2 hydrolysis. It causes the dissociation of the pH PLC delta domain back into the cytosol, represented here in its YFP intensity in the cytosol. Furthermore, this protein, when PKA is inhibited using a specific inhibitor called H89, you can see again the recovery of the cytoplasmic cerulean intensity, and a concomitant, because lipid phosphatases are now away from the substrate, you can see the recovery of HIP2 levels as seen with the pH domain of PLC delta. Thus, we've utilized HIPs to control the enzymatic action in living cells, and this gives us a site-specific control of signaling events. Furthermore, the reversibility, a unique feature among methodologies, existing methodologies, makes it possible to examine the functional effects of switching a signaling event on and off. Thus far, I've talked about two independent projects that I worked on where I've developed a sensor to monitor a signaling event, and in the next project, I've utilized a HIPS strategy to perturb a signaling event. In the third story that I'm going to tell you, we've developed a cyclic AMP manipulation tool to site-specifically generate cyclic AMP signals, and then using that in conjunction with mathematical modeling, we've uncovered the regulation of nuclear PKA. So just a little bit of background about this signaling pathway. The classical cyclic AMP-PKA signaling pathway is such that upon hormone binding, the G protein coupled receptors undergo a conformational change, and this in turn activates G-stimulatory protein, which in turn activates transmembrane ACs. And these enzymes catalyze the conversion of ATP into cyclic AMP. And signal termination of this second messenger is brought about by an enzyme called phosphodiesterases. Once cyclic AMP is produced, it rapidly diffuses through the cell and activates its two major effectors, PKA and EPAC. Cyclic AMP binding to PKA induces a conformational change in its regulatory subunit, and this causes the dissociation of the catalytic subunit from the regulatory subunit, and then it phosphorylates its multiple targets involved in glycogen and steroid metabolism, muscle contraction, and in the nucleus, gene expression. So the nucleus defines a unique signaling compartment where PKA regulates transcription and RNA splicing. And the dogma for cyclic AMP signaling in the nucleus has been that upon activation of the cytosolic pool, the catalytic subunit dissociates into the nucleus and phosphorylates its multiple substrates in the nucleus. However, contrary to this dogma, the existence of a nuclear PKA holoenzyme has been proposed. However, the existence and activation of this enzyme has not been convincingly proven, partly because there is a dearth of tools to manipulate cyclic AMP signal in specific subcellular compartments. So with this aim in mind, we utilized an isoform of adenylyl cyclases called soluble adenylyl cyclase. And it's a G protein independent form of AC, and its means of activation are bicarbonate and calcium. Furthermore, this is insensitive to a drug that activates transmembrane ACs called forskolin. So over here we have tagged soluble AC to different, or targeted soluble AC to different compartments in the cell via localization signals, where the nuclear export signal localizes it to the cytosol, the nuclear localization signal to the nucleus, and then the plasma membrane targeting motif targets it to the plasma membrane. As a marker for expression as well as localization, we've used a fluorescent protein called M-cherry, and it's a red fluorescent protein variant. And we dubbed this method SMICUS, which stands for spatiotemporal manipulation of intracellular cyclic AMP using soluble AC. So